Okay. Hi, everyone. It's great to be here and represent our team and talk about how we can build a human-centered AI virtual cell. And the cell can be considered a fundamental unit of life. And being able to simulate it, I think, has been the dream of biologists ever since Hooke discovered the cells 500 years ago. And we believe that maybe now we're at the point that we can actually do it, because of the very large amounts of biological data that has been generated and can be generated, and also because of AI and the ability to embed and learn from these data modalities. So we believe that this dream of building virtual cell models that are capable of simulating biology is at least closer than it's ever been. And the impact of having a virtual cell model would be enormous. It would accelerate biomedical research at large. Maybe we would even simulate everything before we do the experiments. It would provide advancements in the fundamental understanding of biology and also accelerate the development of drugs. And a very important point at the end there, it would also democratize access to complex biomedical modeling. And this is not an easy task at all. And I want to break it down a little bit because I know that many people here don't have a biological background. So I want to talk about two core fundamental principles of biology that we think are essential to build a virtual cell model. One is, of course, the central dogma, the flow of information in biological systems. So we have our genome, the DNA, that encodes for transcripts, the RNA, that in turn encodes for proteins. So these are all modalities that are basically sequence modalities that we can measure very well at large scales. And while the genome is the blueprint for everything, I do want to point out that the executors of functions are the proteins. The proteins are three-dimensional structures that provide all the structure to the cell. They provide the functions for the cells. They are enzymes, and they're almost always the drug targets too. So it's important to think about all these different molecules in virtual cell models. And this central dogma is the fundamental process for all life, and it provides the molecular basis for how genes are expressed and copied and how mutations can arise and pass down through generations. So it's really, this is what drives evolution. And this evolutionary conservation is something that can be leveraged to really train large AI models, as has been shown by many people already and will be shown by other teams later today. So the AI virtual cell needs to learn the molecular sequence language of life. But then there's another angle that people in this space often tend to forget about that I think is very important. And it's the principle of self-organization. Self-organization refers to the emergence of an overall order in time and space of a system, resulting from the collective interactions of its individual components. So if we think of a human, for example, or any organism, we humans, we have organs with specialized functions. These organs, in turn, are built up by tissues with specialized functions. These tissues are built up by different cell types that interact in a specific way to provide these specialized functions. And we have thousands of different cell types, brain cells, skin cells, that also are specialized. They have a unique set of genes expressed to be able to perform that specialized function. And within the cells, we also have a conserved structures. We have organelles, we have mitochondria, the powerhouse of the cell that produces energy, and we have the nucleus that harbors the DNA, for example. And within these compartments, we have the molecules, like the proteins again, that are executing this function. And this self-organization is key for robustness and adaptability of cellular and organismal organization, and also constitutes a basis for evolution. So we believe that we can use AI to also learn across both these pillars, the kind of sequence, the dogma evolution, and the self-organization evolution. So we claim and believe that the AI virtual cell model needs to be spatially and structurally aware as well. So to kind of summarize what I try to explain there is that the cell is really a three-dimensional functional system. If you compare it to a protein, we can fold proteins now, but the diameter of a cell is 10,000-fold larger than a single protein. So it's a big problem. It's a difficult problem. And a high-fidelity AI virtual cell model must be able to simulate the building blocks, but also their spatial organization and how they interact, because that's how emergent properties arise across scales. So how are we trying to do this? This is a very difficult problem. And it requires working with many different data modalities. So we, in this project, started out by identifying what are the key data modalities that we need in order to build a 3D virtual cell model. And we zoned in on these 4 data modalities, but of course there's even more. Sequence data, structure data, image data, and text data. And of course, this touches upon many different disciplines in biology, and that's why we assembled this fantastic team that I'm very happy to be working with. So we have Jure Leskovich, who's an expert on graph learning and biomedical foundation models. We have myself, that is an expert in image-based cell modeling and proteome systems biology. We have Stephen Quake, who develops sequencing technology and is an expert in sequence-based cell modeling. We have Serena Young-Levy, who's an expert in computer vision and multi-scale modeling. And Russ Altman, who's a specialist in 3D structural protein modeling, drug interaction modeling, and pathway modeling. So we believe that we touch upon all of these 4 pillars with this team. But again, we're not the most important part of our team. Our groups are, and many of them are here in this room. But I just want to show this plot about the students and the postdocs in the lab and all the different Stanford programs and departments that they are within. So we're really spanning many different areas of biology and computer science within our team. So that's been great fun. And over the year, we've just been working on this for a year. We've been meeting every other Friday. And unfortunately, this is the best photo that I had because I always forget to take photos. We're deep in the discussions, and it's been a year where we've learned a lot. And today I'm going to tell you a little bit about what we've done in this first year. So our approach, again, to break it down, well, actually, I can start with this. So what this project started with is that many of us, so me, Jure, Steve, we organized this workshop with lots of people in this space where we talked about how to build the virtual cell with AI. Serena was also there. And that resulted in this perspective paper published in Cell that really touches upon what would a virtual cell be? How could we build it? What is needed from it? What capabilities should it have? It should be able to simulate perturbations. If you treat it with a drug, it should simulate the effect of that drug. And we also talk about what's needed from the community in terms of ability to interact with the virtual cell model, evaluate it. How can we make sure that genetic data is kept private and such? So it's a good read if you're interested in it. I can't walk you through it, but this really was the seed for this project. And then we've kind of shaped our own technical approach based on what we discussed here. So the approach is really to work with multimodal, multi-scale models and joint agentic analysis ecosystems to access these models. Also factual knowledge from biomedical resources. And in the end, we haven't been able to start working with this yet, is application level evaluations and guardrails to safeguard the information in these models too. And in the first year, this is a summary of everything that we've done in the first year. We've actually published 21 papers and developed 7 models, 3 datasets, 6 benchmarks, 3 AI agents, and provided 4 opinion and review articles, just outlining how we think virtual cell models should be built. So I'm obviously not going to walk you through all of these, but I will just provide some highlights from these different aspects. In Sweden, I'm Swedish, I would call it a smorgasbord of what we've done. So I hope you enjoy it. Starting with the sequence models. Models trained on sequence data, again, DNA, RNA, protein, can allow us to represent cell types and states robustly across organs, tissues, and species. So this is where Jure Leskovich and Stephen Quake collaborated to develop the Universal Cell Embedding Model, the UCE model. It's a biologically informed foundation model for single-cell gene expression. So we can measure which genes are expressed in different cell types at large scale using single-cell sequencing. And they actually, as part of this work, they assembled a lot of public single-cell sequencing data sets, actually 300 different data sets, capturing 50 tissues, that's the majority of the tissues in the human body, from 8 species, and in total mapped 1,000 different cell types. So it's the first time they were able to bring all of this into a joint embedding space using this model. And what is also very nice is that UCE can map any cell from any species into this shared universal space. Let's see if this pointer works. So you can see in gray there, we have the data, this universal cell embedding space. And then if you have new data, you're sequencing new cells and you want to know what they are, you can just map them directly onto this universal cell embedding space and understand what they are. And they demonstrated this nicely with novel sequencing data from the African green monkey lymph nodes, for example. But we also know that no model is better than the data that it's trained on and that it's fed with. So in parallel, Stephen Craig's lab has also worked with the Tabula Sapiens Consortium to generate Tabula Sapiens 2.0, a single-cell sequencing resource that is being used by people all over the world. And this resource really doubles the number of cell counts that we have, and it's specifically focused on enabling sex-specific and controlled genetic modeling of cell types and states. So basically, they learned from where they had poor resolution in this previous work to be able to target that type of data generation going forward. Moving to structure models. So models specialized on learning protein structure can advance our understanding of protein-protein interactions, drug-protein interactions, and cellular pathway activity. And this is where Russ Altman is the expert. So for example, this is just an image that I borrowed from another publication. It shows the oncoprotein KRAS. And in the middle plot, you see all the known drugs that are binding to KRAS. They seem to be binding in these hotspots. And then you can also see how known inhibitors bind in those hotspots. So this points towards a great need to be able to know where the binding pockets of proteins are, because then you can do targeted drug design. And this is what Russ's lab set out to do. So they assembled a benchmark of more than 15 million known and predicted pockets. And they also trained this model, HotPocket-NN, that identifies ligand binding pockets on proteins. And it also outperforms existing methods and managed to discover druggable sites on very challenging targets like KRAS, as shown here. So with this model, we can really start to know the structure of proteins and figure out where the interesting pockets and surfaces are where drugs could be designed to bind. And I'll get back to this later. Moving to image models. Image models gives us capabilities to model and simulate biology across scales, including the spatial organization of proteins within the cell, but also how cells are organized in tissues, and so on. It's also very common measurement modality in drug development. So you treat cells with drugs, and then you measure how they respond. You measure their phenotype. So here we collaborated with the Human Protein Atlas, which I'm a co-director of, and used their genome-wide image collection showing protein subcellular localization. So in this image, you see in green different human proteins, and sometimes you can see that they're in the nucleus, sometimes they're in the mitochondria. And as I explained earlier, this means that they're probably involved in different biological processes, and they're definitely not interacting, for example, if they are in different compartments. So we trained a model called Subcell, a vision transformer model using this multitask learning framework, where we introduced a new protein-specific loss that was actually very important. And this made it possible to train this proteome-aware vision foundation models for microscopy data sets. And we've demonstrated that these models outperform the state-of-the-art, the Dino version 2 models that are fine-tuned here even, and many other models on a variety of downstream tasks. It even outperforms a Dino model trained on yeast cells without any fine-tuning on the yeast cells. So it's a very performant model because it learned to look at patterns informed by the proteome. And as I told you earlier, the proteins are the executors of biological function. So that is probably the reason that this model is very performant. So what can we do with this model? Well, we can use it to embed all the protein localizations, so spatial information of where proteins are in the cell from the Human Protein Atlas images. So here, I'm just showing you how we can bring this information into a joint embedding space that shows this is how proteins are distributed in the cell. And at this point, we started to ask ourselves, but what if this information is already captured by sequence models like ESM2, for example, that capture the amino acid sequence of a protein? How similar are these spatial embeddings to molecular sequence embeddings? So this is maybe a bit of a complicated plot, but this is an even more compressed representation of all human proteins embedded in subcell at the top row and clustered at different levels of resolution. In the second row, you have ESM2 embeddings of amino acid of proteins. So that's the sequence embedding of proteins, and also clustered. And in the two lower rows, we have the subcell embeddings, the subcell clusters, colors projected onto the ESM embeddings, and vice versa. And you can see that these two bottom rows, you see no clusters. The colors are all mixed. So this means that the spatial embeddings from subcell have very orthogonal information to the sequence embeddings from ESM2. And this is, of course, very encouraging, because this means that what if we try to join these two embeddings? Can we get better performance when trying to understand the function of genes? So we demonstrate this, we do multimodal embedding of ESM2 and subcell embeddings, and we can show on many different tasks that it outperforms the unimodal embeddings. So to the left here, we're trying to predict protein-protein interactions. And again, this is important because most proteins execute their function by interacting with other proteins. And in the plot here to the left, you can see that the blue bar is the multimodal embedding, and the other two bars are the sequence and the image embeddings. And you can see that the multimodal embedding outperforms the unimodal embeddings by far, more than an additive effect. And even more interesting, the plot to the right, it's a maybe a boring plot, but it's the most interesting plot I'm going to show here. So if we take a biological question like paralogs, that is genes that have a common ancestor, they duplicated somewhere back in evolution, but that are either functionally divergent, so they've evolved to have different functions, or are functionally similar. They duplicated but kept the same function. So if we look at functionally divergent paralogs, the plot, the heat map to the left in the rightmost box there, we can see that ESM, the sequence model, it can't distinguish these two paralogs. They are too similar. But when we add that multimodal embedding, so the spatial information about where these proteins would be, we can see that the multimodal embedding can distinguish the two paralogs. So this demonstrates that cells, when they want to, let's say, use the same gene, the same sequence, but use it to encode many functions, the cells use that spatial access to differentiate that function. Whereas the functionally conserved paralogs are very similar also in the multimodal embedding. So we also tested this model on a task such as predicting gene function, biological process, it's called in the gene ontology database. And we can show that the joint embedding more faithfully reflects gene function. So this is, again, likely because we're joining information about sequence, the central dogma kind of evolutionary pillar that I talked about in the beginning, and spatial location, that self-organization evolutionary pillar. And when we join those two embeddings, we get a better performance in understanding biological functions. And this might sound very basic, but this is the first time this has ever been demonstrated. And we can also use subcell to link it to other types of data, such as protein-protein interaction data. So here we took the images that have information about the global distribution of proteins and joined it with protein-protein interaction data, which is the local distribution of proteins. And again, we can bring it to a joint embedding space. And with this embedding space, we actually built this static model with hierarchical clustering. So this is a model of cellular organization from the cell down to organelles, to smaller interactions, and so far. And it might seem very trivial, but this is the first data-driven representation of the organization of a cell. We have this in gene ontology already, but it's man-made, and it has a lot of intrinsic biases in it. So in this paper that was published in Nature earlier this year, we demonstrated how these simple cell maps that are learned directly from data can be used to gain novel biological insights, such as, well, we resolved these 275 molecular assemblies. We could assign secondary functions to almost 1,000 genes. We could show that you can use it to guide protein-protein interaction modeling, and also use it to identify assemblies that are prone to mutations in pediatric cancers, for example. So again, demonstrating that the value of understanding this self-organization principle of cells. But the image modeling is also important for understanding drug perturbations. So to add back to the structural modeling, that we need to understand the structure of proteins in order to understand how drugs bind to them. And in pharma and academia, people do drug screenings using imaging. So Serena Young-Levy's lab generated this model called CellFlux. It's a flow matching model, a generative model that basically has learned it can predict how would this cell look like if I perturb it with this given drug, or if I knock down this gene. So we can do in silico drug perturbations and generate images of this. So even though this is still early, we're very excited about linking these models. So to get back to my earlier slide, high fidelity AI virtual cell model must be able to simulate the building blocks and how they are organized. And although we're nowhere near a holistic virtual cell model in any way, we feel like we've come far in building these different models to represent the sequence, the structure, how drugs interact with structures, how the cell is organized, and also how the cell organization changes with drug perturbations. So the next phase is, I guess, the exciting phase. It will be multimodal and generative and include lab-in-the-loop settings. So the one pillar that I haven't talked about is, of course, text. of course, we should make use of the vast amount of literature out there. And here we've really focused on benchmarks to start with, because we need good benchmarks in order to evaluate our models. So we've developed multimodal vision text benchmarks that evaluates perception, reasoning, and multimodal alignment across scales, offering a unified framework to measure progress in this biomedical vision, language, understanding, and reasoning. And this is really work led by Serena Young-Levy. And we've developed 3 different benchmarks: Biomedica, Microbench, and MicroVQA. Biomedica has really pulled all image text pairs from PubMed, and it's been downloaded over 50,000 times. So it's really not only used by us, but by many other people. And then we've developed these 2 targeted benchmarks, Microbench and MicroVQA, that are really focused on microscopy images and texts. And with that, of course, this is a smorgasbord of what we've done. It's not a holistic cell model. It's models that are valuable to accelerate biological research that we want to wrap under some kind of an umbrella, and for an accessible virtual cell interface. And here, AI agents allow biologists to interact with complex biological data across the key modalities that I described through natural language. And AI agents also enable users to analyze their own data sets within the context of our AI virtual cell platform, if you will. And this is work led by Jure Leskovic, and he's developed this platform called Biomni, and I will hand over to him.
Thank you. Thank you. So what I want to kind of talk is, as Emma has shown, all this great work, all these great advanced models that basically allow us to build a digital twin of the cell. There are not too many scientists in the world who are actually able to take advantage of these models. So we took this journey to say, okay, how could we make all these models, all this knowledge available to human scientists, and how could we create this human, AI scientist collaboration to speed up the rate of scientific discovery. And we came up with this biological agent or AI scientist that I'll call Biomni. The name comes from 2 words, bio and omni, like everywhere. So, the question is, can we create an AI virtual biologist that makes, this research that we have just seen kind of universally accessible and helps us with automating and speeding up biomedical research? Right? And if we can do something like this, it would have a tremendous impact, because now we can think about biological scientists, human scientists, kind of augmented by AI. You basically can now have an impact of an entire specialized team, right? I think that's very important, because this means that we are making research universally accessible, dramatically reducing barriers, right? If you think about this, there are not too many places in the world like Stanford where you truly have a critical mass of people that are experts in their own areas who can come together and do something amazing together. If you can now augment some of these capabilities with AI, basically we democratize access and speed up science. And the point, as you are going to see, it's not really about replacing, it's about amplifying and extending human scientists' potential. So what we have built is an agent that operates in an environment, right? What is an environment? An environment are all these tools, models, databases, data lakes, and things like that scientists use in their daily lives. So the first step to create this environment with over 250 different, let's say, tools that the agent can use was actually to build another agent who's reading papers across all the fields of biology and extracting what are the tools, what are the methods that scientists are using today. So we integrated this into a single environment where now the agent can basically access all these different tools and all this different knowledge. And the hard part here is that a lot of these models, databases, knowledge bases that scientists are using in their daily lives, they've been made to be usable by humans, but they were not meant to be usable by agents. So there is a lot of work to kind of make these common resources, public resources, agent ready. So now, what is the agent? The agent gets a description of the task. It reasons about how it's going to solve the task. It can use any of these 250 different tools, models, know-how templates, ontologies, and so on, to execute the task in this environment. The agent reasons, it writes a lot of Python code, executes that Python code, analyzes the data, sees the feedback, the output of the Python code, and kind of goes to the next iteration. And beyond just kind of the infrastructure where all this can happen, the natural language agent on top that has a bunch of different sub-agents, there's a lot of emphasis that we put on kind of guardrails, observability, auditability, security, and things like that, right? So kind of the human scientist is really in control. They see the entire trace of thinking, of data analysis, can stop, can influence it, and so on. So we benchmark this quite extensively, and it works really, really well across a number of different problems that, let's say, people or scientists in drug discovery, biomedicine are encountering. Everything from, GWAS causal gene discovery to variant prioritization, perturbation screen design, single-cell RNA-seq annotation, patient gene prioritization, some drug repurposing, rare disease diagnosis, and so on. And beyond this diverse benchmarks that I'm showing you here, we actually have Stanford scientists apply this. And this is one example where they want to do cloning protocols, where they basically want to design wet lab experiments. And they come, this thing here is the description of the task. They say, I have this plasmid for, CRISPR, here's my sequence, I want to design a cloning protocol. The agent comes up with this 7-step analysis protocol, and at the end generates the cloning protocol, right? And if you look at this cloning protocol, it looks like to me like a cooking recipe, right? It's like you do something at this temperature for this long and things like that. But the point is, at the end, they tried it out and it worked. Right? So kind of from the interview from the scientists, they say, look, it would take me 3 years of training and a lot of tool hopping and things like that to design this type of protocol. The agent did it in 10 minutes. I didn't have to, use many different tools, no errors, and I didn't waste any time. So it's very useful for this type of use cases. Here's another example. This is with Mike Snyder's lab here at Stanford, where they basically have very diverse data from human physical activity, continuous glucose monitoring, activity data, food data, and so on. It was about 460 raw Excel files that they dumped into the agent and said, analyze this data, come up with some realistic hypotheses and patterns you find in this data. The agent went on this, like here, 10-step data analysis procedure, created a bunch of plots and visualizations, and at the end came up with scientifically valid hypotheses. Minta, who's the scientist in Snyder Lab, said, while it would take me about 2 to 3 hours per day and several weeks to complete this, the agent has done it in 35 minutes. So this just shows how accessible now these kind of advanced tools become. And we have actually released this into the public as an open source project. And in 3 months, we had nearly 10,000 scientists use it. About 2,300 are using this monthly. We have hundreds of users who are spending significant amount of time out of this, let's say, 60 working days. They are there for 10 plus days. And the top user is basically using it every day, 51 days out of 60 working days in this 3-month period. Overall, we automated more than 60,000 different tasks, which basically equals millions of hours of human time saved. And we see huge interest across, 4,000 different organizations have created accounts and starting to use this. We have also built strong partnerships, with Anthropic as well as other, let's say, nonprofits and players in this biomedicine works, like Ginkgo Bioworks, Tamarind Bio, SCverse, working with PyLab robot, where we can take our agent to actually be connected to the live robot who is doing live experiments kind of in the wet lab, all the way to kind of making other data sets like Sage Bioworks accessible to the rest of the world through this simple agentic interface. And you can see how this really kind of lowers the barrier, because a lot of the scientists, they are not expert PyTorch, Python programmers. They don't have the fanciest GPUs under their desks and things like that, right? So just that enabling them to access these tools, this knowledge, this data in a very easy way makes a huge difference. What I also want to show you is a bit where we think the future is going, right? And could we actually build this and make it much more kind of closed loop, right? Not just here is the task, can you solve it for me, but really, can we generate hypotheses? Can we validate hypotheses, right? And we developed this framework called Popper that basically is based on work of philosopher of science Karl Popper, who basically said scientific progress is pushed forward by constant falsification. So basically, rather than you trying to find evidence for a hypothesis, we are trying to disprove it, and we keep disproving hypotheses until it's disproven, and then we know it's not true, right? So there is this kind of idea of we want to do falsification tests and find refutable evidence to discard the hypothesis. And we have actually built an agent who is using the biomni as a big chunk of it, where kind of the mother hypothesis comes in. The LLM is then generating falsification tests for that mother hypothesis. Biomni executes the falsification test. We get out the p-value, and then we kind of accumulate those p-values. If you would do the proper p-values, it's actually not statistically valid. So we collaborated here with the statistics department and use the concept of e-values that are actually here statistically valid in this case, and you can aggregate them and actually do proper statistically rigorous hypothesis testing with an LLM agent. And what do we see is that basically this agent is as good as human experts, but about 10 times faster. So it's kind of using very similar techniques, very similar statistical tests, very similar falsification tests for falsifying this hypothesis. But when you are looking at kind of the productivity metrics in terms of how fast is it doing things, how many tests, how many lines of code, we see a couple of fold improvement. And this is interesting, right? Because what would this allow us to do is to kind of let this kind of agent in this kind of large-scale databases where maybe we as humans haven't had really time to analyze this data, to go and identify as hypotheses that the current data cannot falsify. And then we can come up with new experiments and really kind of find the set of interesting, plausible, and not yet refuted hypotheses. We are also interested in the question, where is this going? Can we teach these large language models to be good at science? So what I'm showing you here is that actually we can use reinforcement learning-based fine-tuning to build much smaller models, like an 32 billion model that is actually far better than what Claude or GPT-5 are able to do. And this is like a small fraction of the number of parameters of the tuned model versus what the big models are. So what this is showing is that actually we can teach these models to do science in a really good way. And they basically, untrained model, kind of a base language model, the performance basically doubles, and it surpasses the best frontier models that are out there today. So what I wanted to show is that really we think that future of biology is agentic, that these virtual biologists will help speed us biomedical research and help scientists with the help of these advanced tools, models, foundation models from the AI virtual cell, plus this layer on top that makes all these models accessible. We think we can truly speed up the rate of discovery. Thank you.
Thank you.
Thank you. So we'll have Serena and Gary also come up here. And Louise, if you're here, I don't know if you've made it. Yes, perfect.
Yeah. You all can take a seat on these chairs too if you
So just to introduce, so unfortunately Steve and Russ are over Zoom. They weren't able to be here today. So we have Gary from Russ's lab and Louise from Steve's lab.
And we'll start with questions from the selection committee.
So what an exciting set of presentations and ideas. Looks like a lot of progress in 12 months since we last checked in. On the universal cell embeddings, I wasn't clear. Have you actually recognized core eukaryotic findings versus what happens with specializations into across the taxa right now? I mean, do you find some foundational pieces of what makes a eukaryote a eukaryote?
1, 2. So what we are doing with the model is that basically it allows us to, for the first time, kind of truly compare cells coming from different organisms and even from different experiments, because in this kind of biological experiments, the amount of noise is so large that even if you and I take the same cell, take it into the lab, kind of the results we get are very different. So this is what it allows us to do. And now we are having, I would say, a number of different collaborations with biologists to use this as a tool for questions like you are mentioning.
Okay, so let's move on to some specific questions. I mean, for on subcell, I mean, when you build the embeddings, have you looked at where pathologies fit in or single mutations that are well known for proteins?
We have not. Well, actually, what we were able to show, so it's estimated that about 20% of human diseases arise because of mutations that lead to mislocalization of the protein. So we took the set of these known proteins and we tested, can any sequence embedding model predict the mislocalization? None of them could for any of those proteins. So we have a benchmark paper showing that. So we need more development, but we're also starting to generate a new data set where we're doing genetic perturbations of all human genes and reading out all human proteins with spatial resolution.
Really cool.
So with that data set, we'll be able to answer questions like this and understand the regulatory kind of spatial regulatory mechanisms as well.
We'll have more time tomorrow. I could ask 1,000 questions. One more though for you, actually. Why do proteins parallel with the same function spatialize? Is it because of the ribosomes in different areas just happen to put out different variants?
Yeah, the mechanism of this specialization, we don't know. I think that ribosomes might be the answer to it, or localized translations. Maybe there's something about the UTR regions, for example, where these genes are localized that drives different localization. But it's a very unexplored area of biology that we touch upon it in one of the review articles we've written, because we feel like this is a space that more people should work in.
It's just one more question for Yuri and Biomni. When you get a big protocol like that, I would think that many scientists would say, do I take a chance with this generative stochastically generated policy, or do I check this? Have you done any like model cards or failure rates on these protocols? You have a big user base you could do some studies with.
That's a great point. Yeah, we two things, right? Like I think one advantage of this is that actually, it's not only question and a result; it's an entire analysis workflow. So what it means is that, for example, very quickly, the feedback we got from the users was, okay, I'm seeing what the agent is thinking, but I don't understand the code. So then we actually started adding explanations for why is the agent taking the steps it's taking. So now a human scientist gets kind of a complete, kind of almost like a notebook. They can completely verify and say, oh yeah, this is doing what I would do as well. It's according to my standards. I will trust this. Versus, they can stop it and say, hey, you are doing the wrong thing. This is how I would like to do it. And the agent can kind of respond to that. So that's how I would say we address this, because we don't want to be a black box, right? And the beautiful thing with the agent is that you can stop it, you can talk to it, and you can audit the entire trail. It's all there, right? It's easier to audit an agent than it is to audit a postdoc, right? Like, you know what I mean?
So I liked how the multimodal models improved a lot of their results in the graphs you showed, but there still seemed like a big amount for improvement. So I'm wondering, what are the future work of like, how would you get those things to get even better?
Yeah, I can ask briefly and then I'll hand over to Serena. So we believe that this has been established in the basis, and we really need to train these multimodal models, vision language models that learns the proteins and the genes and what's the phenotype of the cell when we look at it, and how do these basically collecting the links from the molecular level to the image level. And we believe that the first step will be a pretty large sequence image model, both from the molecule to cell stage, but also from the cell transcriptome to tissue level. So Serena, maybe you want to add to that.
Yeah, I think just adding on to what Emma is saying, I think in this first year we have a lot of modality-specific models. We have some multimodal models. And I think moving forward, we want to learn a lot of aligned representations between them. We also want to learn very large models that can take both paired, unpaired data, kind of aggregate all of this together, and then make it accessible through these sorts of agentic systems. And so that's kind of what we're looking forward to do. But there's a lot of kind of pairs and links between some portions of data, and that's what we're going to leverage.
And then a second question, really, in the in this work that Yuri talked about, that Biomi agent. I don't know, talking to Eric all the time, I get scared about a bunch of these things. So I'm kind of curious about the guardrails and the work there in terms of avoiding, some evil pathogens and other stuff people creating with your agent.
Evil postdoc.
No, that's a great question. So we do several different things, right? So the first thing we do is, of course, in prompts and in tools, we are very specific what we kind of allow the agent to do and what we allow agent not to do. We are very specific with the selection of tools and the selection of kind of data sets and knowledge that is kind of integrated in the environment. That's one part of the kind of the security. The other thing I would say is whenever people answer questions, whenever you go ask a question, you basically get a fresh machine, right? Because you need a fresh environment. So Biomni is an LLM plus a freshly installed machine with these 250 tools, so that whenever the LLM writes the code, it gets executed here. But then the machine kind of gets wiped out, so nothing remains, so that the data is secure and things like that. So that's, I would say, at several levels how we deal with that. What we are doing now is actually, analyzing the questions, analyzing the interactions, as well as doing a number of user studies.
So, I mean, in the large commercial models, obviously, there's a lot of like red teaming and other stuff. Is that something that needs to occur here if you're using something like Quen, or like what would be the other approaches if, let's say, this was a product? Like, what would need to happen?
That's a great point. So right now, the model we are using is Anthropic, but exactly, you're right. Like proper red teaming is the way to go here.
We have time for one more question.
Really great presentation, super impressive, very exciting results in such a short period of time. I haven't had a chance to read the how to build a virtual cell paper that I just learned today. But in relation to your vision, do you feel like you have all the foundation models built? Like you mentioned, the universal cell embeddings and then cell flux and many other models, and then multimodal cell morphology something. But do they complete the whole picture, or are there missing pieces? And could you articulate what's missing? And also, how does Biomni fit together? I imagine that Biomni can make use of some of these learned models as individual modules, and then you do more research, and then what happens? Is it like the data flywheel?
Okay, so that's multiple questions. So for missing pieces, there are plenty, and many of it is also, for example, protein-protein interaction data would be lovely to have and a model for that, but it's very difficult and expensive to generate such data, so we're very data limited. So I think the models that we have sets us up very, very well. What we don't have is the multimodal models that are like jointly trained and optimized. So we're going to go for that, and maybe what I think might be missing is some of Serena's work on the cryo-electron tomogram, where we all really have this that bridges the spatial resolution with the kind of molecular and structural information. And of course, we're limiting our work also to begin with the scale of the cell to start with, and then link out as well. I personally believe that if we think about virtual organism even, I think that the scales that will be hardest to bridge is actually the subcellular scales, because cells are discrete units, and as soon as we go above cells in scale, it's easier because we have these discrete units. Within cells, there's a lot of dynamics happening. So that temporal axis we don't have covered really well again either, and also we're limited by data there. What was the second question? Was
Biomni.
Oh, Biomni. Yeah. So we didn't have we didn't show that here, but we've been working with Biomni as a kind of wrapper to integrate these models. So for example, we used another model that we haven't published yet, so where we can do generative in-painting of where proteins are in cells. So we used that and connected that to the paralog discovery, for example. So it's basically a wrapper that makes it easy to interact with all these models and chain these models until we have the multimodal representations. But maybe Yuri can add to that.
Yeah, so maybe I can just say, right, like for this AI virtual cell, right, one way maybe that appears is, oh, there are all these kind of separate models, separate modalities, and so on, right? But the vision and kind of, I think the technical breakthrough is that, which we demonstrated now, is that you can kind of learn across the scales of biology, right? If you say, how many molecules do I have? You have a gazillion of molecules. You have, I don't know, several orders of magnitude less cells, but you have 70 patients, right? So you cannot train a model with 70 labeled patients, right? So you need a technique that kind of allows you to jump these physical scales, and even for the patient model, be informed by all the molecules you found in the patient. And the UCE model takes the first step. It takes a step from understanding the molecules and encoding them using molecular foundation models like ESM and so on, and bringing those together at the level of the cell. Now what we've done is also a next level model that now takes kind of individual cells and gives you the kind of embedding or representation of the patient or the donor. That's the vision, and we see, and I would say we have technically de-risked it to show that this is possible, right? That you can basically get even an embedding of the patient that is embedded all the way down to your individual protein mutations.
And I would just say also that the components we've built, I think, our perspective is, key components towards a first useful virtual cell model, where now already given what we have, although there's, filling in all the missing pieces is a community-wide effort. I think that these should allow us to start actually demonstrating and testing utility of these, and especially with the Biomni agent and with these sort of chat and text-based interfaces to allow starting to test that utility in the upcoming years.
And we've already, as written in the report, used, for example, UCE and some models to inform data generation in Tabula sapiens spatial and Tabula sapiens 3 that is being planned right now. So we think that this kind of iterative development of models and data sets will help us to plug those gaps as well.
All right, let's give them a round of applause. Thank you so much.